aerial worms, flatworms, tapeworms, and none of the above. Second question is menstrual cycle, vasopressin, progesterone, FSH, oxytocin. The elephantosis is caused by the Wuchereria bancrofti, that is the filarial worms. Here the elephantosis means when Wuchereria bancrofti or this filarial, these are the nematodes, when it enters in the body due to the mosquito bite, here the elephantosis is occurred. So your answer should be filarial worms. The second is the menstrual cycle. It depends upon that vasopressin, especially the vasopressin is nothing but the vasopressin is this, as well as the progesterone, FSH, and oxytocin. Oxytocin is the birth hormone. FSH, that is the follicle stimulating hormone for that is the follicles are stimulated, progesterone and vasopressin. The vasopressin hormone is not, but the progesterone hormone, menstrual cycle, that is during menstruation, the progesterone level, that is when fertilization takes place, that is by fusion of male and female gamete takes place, progesterone is secreted to maintain the pregnancy. So your answer should be progesterone. This is the progesterone. Look at here. Due to the FSH, the follicle stimulating hormone and estrogen and the progesterone level. This is the estrogen and the progesterone level. Progesterone, that is when to maintain that pregnancy, and here due to the estrogen and progesterone level, it can be pregnancy can be maintained. So the menstruation starts due to the or it depend upon that progesterone. The second question is that the role of Leydig cell gives motility to the sperm, synthesis of testosterone, the nourishment to the sperms, and to undergo the spermatogenesis. So these are the Leydig cells. Here the Leydig cells are present in between that seminal vesicles, the Leydig cells, and it is responsible for secreting the synthesis of testosterone. So your answer should be testosterone. Your answer should be testosterone. Synthesis of testosterone. In ovule, the meiosis occurs in, look at here, in ovule, meiosis occurs in, this is the megaspore mother cell, and here the meiosis is occurred to produce the haploid megaspore. So in ovule, in which part the meiosis is occurred? The meiosis is occurred in a megaspore mother cell. Here due to that the 4 megaspores are produced. Out of that, the 3 which are present in the above, they get degenerate. Then here the sequential 3 successive mitotic divisions takes place. So your answer should be the megaspore. Your answer should be megaspore mother cell. Your answer should be megaspore mother cell. Another question is the test tube baby technique is called test tube baby technique is called as in vitro fertilization. You should remember that whenever it takes place in a laboratory condition, then it is called as the in vitro fertilization. Which of the following type requires the pollinators but results in the genetically similar autogamy? Genetically similar autogamy. So Geitonogamy means it is the transfer of pollen grains to a stigma of a different flowers produced in the same plant, and it is a functionally similar to cross the pollination. It involves the pollinating agent but cannot bring about the genetic variation because it is a self-fertilization. Because it is a cell, suppose this is the plant and here the flower is present and here the flower is present, and the pollination is occurred in between two flower, that is the stalk is the same, the plant is the same, and in the same plant when the pollination takes place due to neighboring flower is called the Geitonogamy. And here genetic variation do not take place because this pollen grains, the genetic variation, that is the genes are same, genotypes are the same in both plants, in both flower, and that's why the answer is Geitonogamy. Answer is Geitonogamy. Okay. The asexual process replaced by sexual method is known as apomixis. Okay, the asexual method process replaced by sexual method is known as apomixis. Then if diploid chromosome number in a flower plant is 12, then which of the following will have the 6th chromosome? Whether in endosperm, leaf cell, cotyledon, or the synergids. So you should know that twice n number chromosomes are present. Suppose in that plant the 2n number of the chromosomes are present. Suppose the 36 chromosome. Okay. Then here the leaf also contains the 36 chromosome. So the leaf is not answer. Endosperm, this is the endosperm, that is the primary endosperm, it is a triplet, 3n, that is 3n number of chromosomes are the triple one. In cotyledon also the 2n number of the present, that is diploid, cotyledon is also diploid. And in synergid, this is the synergid, and synergid having it is a haploid, it having a single set of chromosome. So what is our question? If diploid chromosome number in the flowering plant is 12, that is the chromosome number in that plant is the 12. So the leaf also contains the 12 chromosome number. Here in the endosperm, how many chromosome number will be there? Endosperm is the triploid, that is the thrice n chromosomes are present. So the 12 plus 6, 18 chromosome numbers are present in the endosperm. In cotyledon is also twice n number. It is as the same like as the plant. So the cotyledon, it also contains the 12 chromosome. And synergid contain 6 chromosomes. So the following will have the 6 chromosome. Which one of the following will have the 6 chromosome? Your answer should be synergid. Your answer should be synergid. Okay. Then the opening of coronary sinus is guarded by. This is the coronary sinus. It is guarded by tricuspid walls, bicuspid walls, sebaceous wall, semilunar wall. Tricuspid wall and the bicuspid wall, they are present in between that ventricle and the atria and the ventricle. Okay, tricuspid and bicuspid walls. Semilunar walls, they are present in aorta, and sebaceous walls are present in this coronary sinus. So your answer is sebaceous wall. Your answer should be sebaceous wall. Then Another question is, in between the two layers of serous pericardium, there is a space called as the cranial cavity, abdominal cavity, thoracic cavity, and pericardial cavity. Cranial cavity in brain, abdominal cavity, you know that, thoracic cavity, you also know that, and pericardial cavity. The pericardial cavity, this is the pericardial cavity, that is the outer layer of heart, is a heart wall. The pericardial cavity, this is the pericardial space, and this is the pericardial cavity. So your answer should be in between two layers of serous pericardium. This is the serous pericardium. In between that, pericardial cavity is present. So your answer should be pericardial cavity. What is the name of tiny sacs in the lungs? The options are bronchiole, ravioli, alveoli, bronchs. So these are the alveoli. So these are the tiny sacs present in that lungs, and your answer is the alveoli. Which of the following is the true for the golden rice? It is a vitamin A enriched with the gene from daffodilly. It is pest resistant with gene from Bacillus thuringiensis. It is a drought tolerant developed using agrobacterium. It is a yellow grain because of the gold. So your answer should be true for the golden rice. Golden rice, it is necessary for the vitamin A enriched with gene from daffodilly. Okay, a golden rice. So your answer should be A. It is a vitamin A enriched with gene. The golden rice, you know that the golden rice, here the golden rice, you generally the rice which having a carbohydrate, but this is the variety in which gene is introduced from daffodilly plant. Irvinia gene is introduced, and this Irvinia gene which is necessary for the to enrich, or it produces the vitamin A. Okay. Which one of the following equipment is essential required for growing the microbes in a large scale for industrial production of enzyme? So which one is that? Bioreactor. Your answer should be the bioreactor. The correct order of step in a polymerase chain reaction. Look at here, which are the different steps. This is the PCR machine, polymerase chain reaction. First step is the denaturation. Second step is the annealing. Third step is the replication of DNA. Okay, so that is, it is also called as the extension. So first step is the denaturation. Denaturation means due to the heat near about 90 to 95 degrees Celsius, here two DNA strands they get separate. Annealing means here the RNA forward RNA primer and the backward RNA primer, it is joined on the single strands, is called as the annealing. And extension means here the nucleotide, oligonucleotide, they get attached to that RNA primer and two copies are produced. This is called as the extension. Okay, so these are the different steps which are related in the PCR technique. So your answer should be the denaturation, extension, annealing, annealing, extension, denaturation, or extension, denaturation, annealing, or denaturation, annealing, and the extension. So the option D is the correct. The process of copying genetic information from one strand of the DNA to the RNA is termed as replication, transcription, translation, and reverse transcription. Replication means from a single single DNA strand, two new strands are produced is called the replication. Transcription, from messenger RNA, from DNA, the messenger RNA is synthesized is called the transcription. And the translation means the message which is present on the messenger RNA, which is translated by the tRNA, is called the translation process. And reverse transcription means here from DNA the RNA is synthesized again, and from RNA again back the DNA is synthesized is called the reverse transcription. So your answer should be, so this is the transcription process from DNA strand. This is the DNA strand. From this DNA the RNA is synthesized, messenger RNA is synthesized, is called the transcription process. The copy in a written form is called the transcription. Then the second question is, which of the following phenomenon was experimentally proved by the Michelson and Stahl? The transformation, transduction, semi-conservative DNA replication, and central dogma. So the Michelson and Stahl experimentally proved the semi-conservative method of that DNA. This is the replication of DNA. That is, semi-conservative means one strand is the new and another strand is the old. Suppose this is the one old strand. Here the replication takes place, and due to that replication, two new strands are produced. Out of that, here the one strand in one DNA, the one strand is the old and the another strand which is newly synthesized. That is the semi-conservative. One strand is conserved. That's why it is called the semi-conservative method, and it is proved with the help of isotope. That is nitrogen isotopes, 15 nitrogen and 14 nitrogen. So this is proved by the Michelson and Stahl. So your answer should be semi-conservative DNA replication. Which RNA carriers in the amino acid forms the amino acid pool to messenger RNA during protein synthesis? Which RNA carrier carries the amino acid from the amino acid pool to messenger RNA during protein synthesis? Which, look at here, this one is the tRNA. This tRNA, it carries amino acids. It carries the amino acids toward the messenger RNA during the protein synthesis. So your answer should be tRNA. Select the incorrectly matched pair. Initiation codon AUG GUG. Yes, it is correct. Stop codon UAA UAG UGA. It is also correct. Methionine AUG. Yes, AUG code for the methionine. And anticodons messenger RNA. No, anticodons are present. These are the anticodons. They are present on the tRNA. So the incorrect match pair is the D, anticodon messenger RNA. The substance produced by cell in a viral infection that can protect other cells from further infection is which one? Interferon, histamine, serotonin, colostrum, interferon, and histamine. A substance produced by cell in a viral infection, especially in a viral infection, interferon works. So your answer should be interferon. Okay. The antibodies present in the colostrum which protect the newborn from certain diseases is of. You know that in the colostrum, generally immunoglobulin A type of antibodies are present. So your answer should be B.